Hello. I want to tell you a little bit about our implementation of Phage Lab. So we do this as a introductory biology lab. It's attached to the first class that students take as freshmen traditionally. It's not the only section. We have what we call traditional lab, and they do more cookbook type labs like students are apt to do in those classes. But we have a special section, and it is the Phage Lab. We let students opt into that. Admissions emails them when they're incoming students, tells them that there's this special section that they could be in. It's in the mornings from 8 to 10 on Tuesdays and Thursdays. So it's actually an hour longer than traditional lab is, which meets on afternoon for 3 hours. But a number of students opt into this. We're not sure if it's just the times that attract them. Some of them, I think, have sports and want to go to practice in the afternoons and opt into the morning lab, maybe for that reason. We're hoping some of them just are interested in the research aspects of Phage Lab, but we're not completely positive. And we usually get about a dozen in that section. And so we've had 2 faculty in there teaching, and that's nice actually because our gas jets are a bit limiting. We have 8 of them set up in one laboratory space that we use, and then the next door over we have 4 more gas jets. And so a number of the students end up going next door when they are actually doing their wet Phage Lab. And it's nice to have the 2 instructors because then one of them can be in that room and one can be in the main room. We start with a direct isolation attempt, and if that fails, then we go on to enriched. Usually a couple of them get something by direct out of 12 students. So we have those students continue on with their direct, and we have the other students start a enriched. And again, all we need are 2 phages at the very end of this. So we're not that worried if some of them never get a phage. If they want to keep trying direct for a while, we're happy with that. I guess our cutoff is maybe around the middle of the semester where we're like, hey, you really need to get a phage to start working with. We also will have them work on another phage that a different student found and help purify it and get a high titer on it. And so there's a variety of things that we can do. We try not to worry too much. I mean, if you're focused on they've got to get a phage, they got to get a phage, that's a little stressful. But again, if it's the second half of the semester, then by that time, probably most people have gotten a phage if they're going to, and we need to do the work to get it purified. So again, we start with a direct, we move on to enriched. We don't worry if all students don't find something, but we tend to have them work in teams in non-COVID years to where then they maybe have a partner and they're trying to work through the steps together. It has worked out by the end of the semester. We usually have something to send in. And so I guess the advice I would give is be flexible, keep an eye on time, but don't constantly worry about it. And usually things will sort themselves out and students will get what they need in the end. So again, our targeted population are incoming freshmen, typically biology majors, but we don't require that. And so we have some exercise science majors in there that have to take the intro bio course. We might have biochem majors. It varies. And again, we usually start with about a dozen students in that section. So what challenges have we faced? contamination usually. And so when we've been talking in our meetings, I try to keep hitting. If you have contamination in your bacteria, you're going to spin your wheels for a very long time and not get good results. So that is something constantly to monitor to see if anything's growing in the media and buffers that you're using to subculture, to try to do all that you can, sterile technique, impress this on the students, but again, not to get contamination in there. If you have pure cultures, you're pretty much guaranteed that things are going to work out. Again, we're flexible, and so we can shift directions. We basically evaluate at any given lab meeting, hey, what is everybody doing? What direction are they going? How is that going? And at the beginning of the semester, all the students are doing the same things, but very soon students are doing quite different things because they're at different stages along the cycle there. And that's fine too. That it helps having the two professors, and we can say, oh, okay, this team over here is purifying DNA. This one is still trying to get a phage. This one is trying to get a high titer. And so pretty soon all the groups are doing at different stages along there. And as long as somebody gets to the very end stage before the end of the semester, we're pretty happy. So again, we have had pretty good luck. We just try to roll with the punches and talk about things. If we're having contamination problems, sit there and try to figure out where the problem might be and talk to the students about the methods. You have to keep going over them. Again, they tend to forget, they tend to get sloppy sometimes, but we keep emphasizing the need for good laboratory technique and to look back over the protocols to make sure you know what you're doing that day, and to work hard. Often, I think I've mentioned, we have them come back in later in the day. So if they flooded some plates, it's not really ideal for them to come back 48 hours later when lab is starting again. And so we tell them, come in, take the lysate off, filter it, put it in the refrigerator. It'll be ready for the next day. But we have the lab open most of the time. And if not, we can open it up for them and they can work a little bit out of the set hours in order to get success that they need. And so again, the students have to be willing to maybe come in at different times and complete the work that they started, because things don't always fit nicely into these little time periods that we've set off. So hopefully that's helpful. If I can answer any other questions out there, then you can drop me an email or something. But just have fun, enjoy it, and get some phage.